you spend enough time and you're careful enough and you're observant enough and you notice some little things that maybe you wouldn't have noticed the first time around, then I think the coincidence will happen and you will be given luck. When I came to grad school, I actually wanted to study monkeys and their visual system. But I quickly learned that it's incredibly complicated, still, nearly as complicated as the human brain.
Botla couldn't move forward without a simpler model. And that's when a friend mentioned the lab of MIT professor Bob Horvitz.
He told me for the first time about C. elegans, the nematodes.
C. elegans is a beautiful animal. One has to just fall in love with it at first sight. And as an experimental organism, it is unparalleled in the animal world. You've got small size, rapid growth, wonderful genetics, and you have simplicity. There are 959 cells in its body.
And I was intrigued because they had very simple brains, 302 neurons, and it seemed like a simple system that you could really try to understand.
At first, Botla wanted to see if the worms could learn. To find out if they could learn to connect a smell with an unpleasant experience, he would give the worms a scent.
And then I would shine light on them, because when you shine bright blue light onto the worms, they run away, they avoid.
While doing this experiment, Botla noticed something strange. It had to do with the worm's feeding organ, called the pharynx.
So normally there's this motion back and forth in their head that's them feeding. And that just stopped. And then they avoided. And the postdoc in the lab was like, "Did you see that?" I was like, "Yeah, so?" And he was like, that's interesting. And I'm like, "It is?" And I kept doing my learning experiments. They never worked. And then 6 or 9 months later, I was like, "Oh, there was that interesting thing I observed. Maybe I should go back and study it a little more."
Botla wondered why light would make the worms stop feeding.
It's not like when you shine a bright light on us, we drop what we're doing and stop eating.
It was also a mystery how the worms sensed light at all. C. elegans doesn't have eyes, and what's more,
the worm didn't contain any proteins that were capable of responding to light directly.
Botla began to investigate. He learned that other researchers had found a gene that was linked to the worm running away from light. But here's what's strange. This gene gave instructions for making a taste receptor. These proteins normally detect chemicals in the environment.
The field was sort of confused. Why is a taste sensor involved in light?
Botla wondered if this link between taste and light could explain what he had seen. He wanted to take a closer look at the worm's other taste receptors. Fortunately, C. elegans' DNA is easily manipulated. Botla modified the DNA of individual worms, shutting off taste receptors one by one. And sure enough, he found that one told the worm to stop feeding when light came on. This gave Botla an idea.
If the light was actually generating something that would be harmful if the worm took it in, then that might be an explanation.
Botla tested his idea by giving the worms the oxidizer hydrogen peroxide. This is part of a class of chemicals that can damage DNA and proteins, and, under the right circumstances, can be generated by light.
And I went to drop it on the worm, and even before I was able to do that, the worm stopped feeding and avoided, instantly. It's exactly, for the first time I'd seen something else other than light cause the exact same avoidance and stopping of feeding.
Was the worm's reaction to light actually a response to tasting chemicals? If this were true, the worm's nervous system should respond to hydrogen peroxide and light in the same way. To see which cells controlled both responses, Botla used a tiny laser to kill each neuron in the worm's pharynx, one at a time.
Every other thing I did seemed to reinforce the same neurons that were involved, that responded to light. They also responded to this odor of hydrogen peroxide. We can't even smell hydrogen peroxide, so this was sort of an extraordinary sense that the worm had, that humans lacked. C. elegans tasting light, I think, is the clearest way to summarize what I've discovered.
This is more than a curiosity. This is a new insight into how the worm navigates its environment.
If something that is toxic is generated by light, the worm is smart enough not to just swallow this toxic substance. And probably what it's doing is it swallows it, tastes this oxidant, says, yuck, this tastes awful, and I'm not going to put that in me.
If Horvitz is right about the worm's discerning palate, that leads directly to new questions. What else can the worm taste? And how exactly does light generate these chemical cues?
It's just amazing. I mean, that simple bit of life is so complex, and we're still at the very early stages of trying to figure out how it all works.
C. elegans is a little human being in disguise. The biology of C. elegans shares so many features with the biology of humans that we have learned many things and can anticipate learning many new things by studying organisms like C. elegans. There is a universality of biology, of genes, of gene pathways, and of genetic and molecular mechanisms that is conserved amongst organisms that are as diverse as a bacterium or a yeast cell or a mouse or a tree or a person or a worm.